Yeah, so can you guys see the screen?
Yes.
Okay. So yeah, I'll just show you. Can you guys see this table?
Yes.
Yeah. So what basically, I'll just update on what we have done so far. So we had taken the raw read, the FASTQ files, which had your reads which came out of the sequencer, along with the quality scores. And we first saw how our data looks like, and then depending on if the quality is good or bad, we proceeded in that direction. So the first thing that we did was we did fast QC, where we were checking how the reads were, how the quality of the reads were, and then we used this software called as BWA. So that uses the Burrows-Wheeler transform algorithm that we had discussed in the last theory class. So that would then help you to align your reads against a given reference genome, and you generated something called as a SAM file, which is the sequence alignment map, which gives you the leftmost coordinate of all your reads that would have mapped to the reference genome. And then that SAM file is sort of your go-to file for whatever analysis you are going to do downstream of it. And then we did a couple of file format conversions. We converted the SAM into BAM, which is just the binary form of SAM. We sorted it, indexed it, and then we saw how many reads mapped. So we did something called as a flag stat, which gives you how many reads that you had. So for example, if you had 1,000 reads, out of these 1,000 reads with all your filters that you had given, how many reads actually mapped to the genome. So let's say from 1,000 you got 900 mapped, and the 100 reads, the rest of the 100 reads did not map to the reference genome. And after mapping, what the only step that is now remaining, which Nitish will complete in the next class, is you have to just assign wherever these reads are into particular bins. So for example, if now I tell you that there are 900 reads that map to your genome, how many of these then map to a particular gene? So now what you're looking over here is a table, so which we will cover in the next class. Uh, this table, the columns over here are the sample ID. So these are the samples that you had that we had we've seen how to download it from the databases. So this is SRR ID 1023811. So this complete column, the first column, the second column rather, is the sample one. Say a third column is sample 2, sample 3, sample 4, sample 5. So we are going to generate this matrix where each row is one gene. So what this first row means is that the gene ID WP003400253, which usually is a DNA A gene, is having 7,622 reads that mapped in this sample. Then 9,073 reads that mapped in this sample, and so on and so forth. So you will, so in, so this is Mycobacterium tuberculosis. If you remember the reference genome that we had downloaded, there are 4,058 genes that are present in this particular bacteria. So for each of these reads across all our 8 samples, we are going to assign, or we are going to segregate whatever total reads that mapped in our sample in these bins of different genes. Do you guys understand? Hello.
Can you repeat the last part?
So what I'm saying is that we are now generating this matrix. So in the last class, we had done these BAM conversions, right? We had generated the SAM file. We had then converted it into the BAM file, which is just the binary version, nothing else.
Yeah.
And then we had created these.
So Mohak, can you just zoom in because everything is too small, at least for me. The
Okay.
text is very small.
Okay. Can you see it now?
Better.
Yeah, much better, much better.
Yeah. So then basically what you want to arrive at is a matrix, which Nitish will do it in the next class. It's just one step. So what basically we are doing is, as I told you, let's imagine that you had 1,000 reads that map to your genome after the BWA, the alignment that you did, and now all you want to then further see that if out of these 1,000 reads that had mapped to your genome, how many of these are falling in a region that codes for gene A versus gene B, C, D, and so on. So all you're doing is you're just organizing your data, going from a generic mapping of across the whole genome to the locus of each particular gene. So once you will do that for all the samples, so over here the columns represent the samples. So we have, if you remember, we had 4 conditions, right? One was glycerol and pyruvate, and within glycerol and pyruvate we had different pH. The paper, if you remember, that shared in the beginning. So, and for each condition we had 2 replicates. So this is replicate 1 and 2. This is 3 and 4. So they belong to the other condition. This is the 3rd condition, and this is the 4th condition. So for each of these samples, what we are doing is for each gene, because all of these samples are coming from Mycobacterium tuberculosis reference genome that we had downloaded the first time. So for each of these samples, we are going to segregate the reads in different gene loci. So this is gene 1. So we are representing this gene with gene ID WP0034 and so on. This also you had downloaded, if you remember, the GFF file that we had done in the first class for the practical. So for each of these genes, you will get these reads, which are now your raw read counts. And remember, RNA-seq is a read counting problem. So the number of reads that are assigned to a particular gene at the end of this exercise would correspond to how much that gene is being expressed in the samples. Are you guys with me?
Yeah. Yes.
Okay. So then we are going to start with what do you then do after you get this matrix of this sort, where you have columns of samples and rows of the gene IDs. So we'll start with something called as a normalization. Now I'll give you the motivation behind why what is normalization and why one wants to do a normalization. So right now what we saw, the matrix is a raw read count matrix. So that means directly you have just assigned whatever reads were mapping to the genome to different gene locus. Now the raw read count of, let's say, a given gene, let's say we take the first gene that we saw, the first row that we saw. Let's just talk about it for now. So the raw read count of a gene could be a result of so many things. So what you're seeing over here in the green is the thing that you want to arrive at. So if the whole reason why you did this exercise of assigning the read counts to particular genes after mapping was to get to this abundance. But there are many other factors which can make you think that this is the true abundance. But in reality, it would be the case that these are actually overshadowing the real abundance. So what you want to do is you want to take care of these confounding factors and apply some sort of a normalization and reach to the true abundance. So I'll explain to you what I mean by taking all of these examples one by one. So first, let's talk about the gene length bias. Now imagine there are 2 genes within the same sample. So this is gene A and this is gene B. Now gene A is of length 2000 base pairs and gene B is of length 1000 base pairs. Now when you did the mapping through the Burrows-Wheeler transform, let's say I tell you that there are 15 reads that you got were mapping to the gene A, and there were 7 reads that were mapping to gene B. Now you might think that this could mean that the expression of gene A is higher than expression of gene B, just because there are 15 reads mapping to gene A and there are only 7 reads mapping to gene B. Now at this point, the assumption is that all the other factors are constant, and the goal is to reach to the true abundance of the gene. So that's what our main goal is. But what when you're reading out this reads mapping of 15 and reads mapping of 7, what you are not doing at this step is not taking into account that the probability of reads coming from a longer gene would be anyway more as compared to a gene which is shorter in length, just by probability. So what then we would want to do is normalize for this gene length and see if after normalization the read count that we get, is there a difference in that or not. So when I normalize for the gene length, what I get is that in gene A there are 7.5 reads per 1000 base pairs, and in gene B there are 7 reads a mapping per 1000 base pairs. So after the normalization, what we see is that in actuality there is, or in reality there is no difference in the expression of gene A and gene B in that sample. But before normalization, it appeared that uh, gene A could be expressing more as compared to gene B. Do you guys get this point of the gene length bias?
Yes.
Yeah. So basically that's the gene length bias, and that's how you rectify for this.
But
Um,
one should also mention for that for normal differential expression type things, this is not required, right? Because you're comparing gene A in sample X versus gene A in sample Y, and
yes.
so that the gene length automatically gets cancelled out. And the other issue is that there seems to be some empirical evidence that for some strange reason that we probably don't know, um, this gene length thing doesn't seem to necessarily scale linearly. So
Yeah.
it's not entirely clear that even if you divide by the gene length that you have an effective normalization. But the good thing is for the standard application of differential expression type thing, it anyway doesn't matter.
Yeah.
Ashwin, I didn't get it. When is it not needed?
Because when you're doing a standard differential expression analysis, which is where this will eventually go, is you're going to compare the expression of gene A in condition 1 versus the same gene A in condition 2. So anyway, the length is cancelled out.
Oh, okay.
So that's the most standard thing, and most of the normalization and the differential expression methods are geared towards that. So any non-linearities that might arise in this gene length versus expression relationship is not really relevant for those standard analyses.
Yeah.
Okay.
Okay. So the next factor we are going to look at is the library size bias. So again, now let's take a hypothetical condition where in this case you have the same gene, now gene A. So the length will be the same. That's the assumption. And all the other factors. So till now we have just seen the gene length. So let's assume gene length is a constant. And again, the aim is to reach to the true abundance of the gene. So now in this condition, the first condition, it's a wild type, and the library size or the sequencing depth that we had with respect to this particular condition, wild type, was 5,000. So there are 5,000 total reads that were that you had gotten from the sample sequencing. And let's say now you get that there are 15 reads that are mapping to the locus of gene A. Now, there is another condition, mutant. Again, the gene length would be the same because it's the same gene. Now, the library size, let's say I tell you, is 10,000. Um, and you see that the number of reads that are mapping to gene A are 30. So if you don't take the library size into account as of now, you might again think that gene A is getting overexpressed in condition the mutant in the mutant as compared to gene A in the wild type, which has just 15 reads which are mapping to it. But when you take the library size into account and you normalize for it as you did for gene length,
Not,
what you see, yeah,
yeah. The library sizes, including the when you say library size is 5,000, it's 5,000 unique reads, or is it including the repetitions?
That's what we discussed, right? So the filter that we applied last time when we were converting the SAM to BAM, the mapping quality filter. So if you have a mapping quality of 0, that means it's multiply mapped. So in RNA-seq, you then really have to take a call. Ideally, it should be uniquely mapped reads, but then things like, at least in eukaryotes, you have things like isoforms. So same read can be coming from different isoforms as well. So that you have to see. So right now, let's assume that these are all uniquely mapped reads. So when we say library size of 5,000, it is a cumulative of the reads mapping to every position in the genome, not just gene A, but gene B, gene C, or and so on and so forth. So uh, out of these 5,000, you have just 15 reads mapping to gene A in this condition and 30 reads mapping to gene A in the mutant condition. Is that clear?
Yeah.
Yeah. So when you normalize for the library size, what you end up seeing is that, oh, there are 3 reads per 1,000 base pairs in wild type and in mutant both. So therefore, there is no real difference in the expression of gene A in both the conditions. So people came up with this one scaling method by including the library size and the gene length bias. So this is what we call as the read sizes for library size. You have on the in the numerator you have the reads per transcript. So in this case, 15 would be that number. So 15 in wild type condition gene A would be the reads per transcript, and then you divide it by this whole expression, which is the transcript length. So the length of this gene, which is let's say 2,000 base pairs, is uh, the transcript length. So you divide it by that, and you also divide it by the total number of reads. And just to normalize it to a number that can be compared, you then further take per base, kilo base, because the average length of the genome is the gene in bacteria is 1,000 base pairs. And then you also divide the total number of reads by 1 million to scale it to a factor that can be comparable. So all you're doing in this expression is normalizing the reads that you're getting, the raw reads that you're getting, with the length of the gene and the total number of reads, or your sequencing depth. There are other measures as well, like FPKM, which is fragments per kilobase per million reads. So fragments is just a term that you use when you have a paired end data. Then you also have transcripts per million, and you have counts per million. So for example, for counts per million, this expression would reduce down to reads per transcript, and now you're just normalizing for the sequencing depth, so you get total reads upon 1 million. Now, RPKM, as Ashwin already mentioned, is not suitable for inter-sample comparisons. Now we'll get to, so one of the points that he mentioned already was that the length of the gene would remain the same across 2 samples. So if you have to compare gene A in sample A and sample B, or sample X and sample Y, uh, for all practical reasons, the length would remain the same for gene A. So so really, you don't need to take that factor into account because you're comparing the same thing across 2 conditions. But other than that, there is one more reason why RPKM does not fare well when you are doing inter-sample comparisons, for example, wild type and mutant. So I'll just give you one example for that. So let's imagine that you have RPKM of gene A that you get in sample X is 5.4, and the total RPKM, which is normalized with the normalized reads of the sample, so across all the genes after normalization, is let's say this number. Now 100,000. So then a gene A RPKM in sample Y is again 5.4, and the total RPKM in this case is 200,000. So what you might, when you just look at this normalized RPKM value after so-called normalization, you might think that the expression of gene A is the same in sample X and sample Y. But you are also not taking into consideration the total RPKM normalized read, which could differ. So the proportion of gene A in a sample could be different from its proportion in the other sample, given the even after the RPKM being the same. So the biggest assumption that RPKM takes is that the composition of RNA in each cell is the same, and that is something which it does not account for. So let's look at what this particular problem is, that the RNA composition of each cell is the same. Now imagine that you have two cell types. Let's say the red one over here is the wild type and this is the mutant. And I tell you that there are three kinds of genes that are being expressed in both the cells. So now I am telling you the truth right now. So let's imagine we play God and we can precisely know that from each cell how many number of reads would you get for each particular gene. So this is the black gene, this is the red gene, and this is the blue gene. So these are three different kinds of genes. And now let's say I tell you that there are 12 reads that are coming out of gene A, and this is the exact number that you somehow were able to know. And there is one read that is coming out of gene B, the red gene, and there are three reads that are coming out of the blue gene. Now in the wild type, there are three reads that were being expressed in or the three transcripts that were being expressed in gene A, one transcript in gene B, and three transcripts in gene C. Now this is the reality. So these are the exact number of transcripts in both of these cells for each of these genes that are being expressed. Now if I tell you, and this is basically what happens during the RNA sequencing as well, that you do not end up sequencing each and every transcript. So it's a matter of probability. So what I mean by that is, now if I tell you to close your eyes and I ask you to pick up 3 sticks from this cell and this cell, could anybody tell me in this cell which are the reads that you are more likely to pick up? So if I tell you to pick up 3 sticks over here or 3 reads over here, which reads would you end up picking up more than others?
The black one.
Yes, yes. So the black one. So just because the number of transcripts were much more, so in your final sample you might end up picking up black transcripts more than the other. So what it might end up looking like is that, so before that, so over here now, once you know this reality, you also know that the gene A is being overexpressed because there are 12 transcripts present here. There are only 3 transcripts present here, but gene B and gene C have the same number of transcripts. So there is no differential expression difference in gene B and gene C. The only difference in the expression is in gene A in both the cell types. But when you do that exercise of picking up three reads at random, what it might appear is that there is a differential expression of even gene B. So you will not pick up any red read count in the wild type, but you still might able to pick up the red read count in the second sample. So in reality, when there was only a difference of gene A, now it seems like there is a differential expression of gene A and gene B. Is this point clear?
Yes.
Yeah. So basically what we say is that usually the highly expressed genes end up monopolizing the whole sample. And so we need to take care of this RNA composition bias as well, which RPKM was not taking care of. So we'll move to a more formal way of understanding this, so that this will tie in well when I will explain you the algorithm that HR uses. Now imagine again, so this is the reality table and this is what you get from your sequencing. So in reality, you know that the total RNA composition in wild type is 2000 and the total RNA composition in the mutant is 1000, in such a way that there are 500 transcripts being produced by gene A, 500 transcripts being produced by gene B, 500 by C and 500 by D. But gene A and B are differentially expressed in reality, whereas C and D are not differentially expressed. So there are no read counts for or no transcripts being produced by gene A and B in the mutant. So it's being highly expressed in the wild type, whereas the expression of C and D does not change in both the conditions. This is the reality. When you do the sequencing, and let's say you wanted the library size to be the same. So let's keep the library size same as of now. So so just to make up for these reads, you will be uh, over sequencing these reads coming from C and D. So what now might happen is that you might feel that there is a differential expression of gene A, B, C, D, all the 4 genes across wild type and mutant. Remember, the library size is the same, so to make up for that library size, you might end up oversampling for the reads from C and D as well. So it might appear that C and D reads are also differentially expressed uh, in addition to A and B, but in reality we know that this was not the case. Only A and B were being differentially expressed. So this is how if I'll share with you the paper that the HR paper that we talk about. This is the example that the kind of sense that they are trying to make, that this sort of thing can come from RNA composition bias, because your highly expressed genes can be monopolizing the genome. So, but basically what this means is what we discussed over here.
Uh, Mok, how is the oversampling? What would be the case where the oversampling happens?
So now you see in gene C and D, right? I told you that your library size is 2000.
Yeah.
So you might end up, uh, now to make up to the 2000, you will be getting more reads from C and D. Because remember, you don't know the original composition of RNA in the cell. Whatever you are, the only truth that you know of is based on whatever sequencing you do. So depending on that, when you do this exercise, again, you don't have a handle on precisely the amount of RNA that is coming from one cell. So you will be getting a pool of cells, right? And then again, the highly expressed genes, when you will take the highly expressed genes, those genes will end up being in the sample more as compared to the other genes. So whatever these reads could have gone to the other genes, now they are all concentrated on C and D. And
I see.
therefore now it can make it look as if these two genes are also differentially expressed as compared to in the wild type.
Okay.
Yeah. So so then coming back to this thing that we started with. So as we discussed, raw read count could be a result of gene length, library size, RNA composition bias, and of course the abundance, which is what we want to get at. So we need to apply for normalization such that we take care of these confounding factors um, and reach to the real abundance. So the software that we are going to use, that you had already downloaded, is the HR software, and it uses something called as a trimmed mean of m values. We'll come to that, what exactly this TMM normalization is. By the way, I would recommend you guys to go and read this paper. So this is the first paper that came in this direction, which talks about HR, and the whole suite of the whole package that we are going to use from now on in R will be from Agile. So just go through this. It explains what I'm going to explain in this the whole series. So Agile normalizes for sequencing depth and RNA composition bias. As we discussed, these are intersample confounding factors which you need to take care of. But it leaves on factors other than these two, which are like it could be gene length, GC bias, and so on. Like Ashwin already mentioned, because the length of the gene would be the same across two samples. So when you're comparing the same gene across two samples, you might not want to express the normalize for the gene. Again, the assumption over here is that the length of the gene did not change in both the conditions. So you really need to know your sample before you apply these normalizations. So HR only normalizes for sequencing depth and an RNA composition bias. Uh, and so therefore, we could think of CPM rather than RPKM in this direction because it takes care of the sequencing depth. And we'll come to how RNA composition bias is taken care of. Now, so if anybody has done a QRT-PCR, could anybody tell me how do you? I'm sure you also normalize in QRT-PCR. Also, you normalize whatever expression you get. So, how do you usually normalize in a QRT-PCR when you do your experiments to get to the real expression of a particular gene? if somebody has done a QRT-PCR. Has anybody done a QRT-PCR?
No.
Okay.
No.
Then I will try to explain that along this when I will be explaining the whole normalization process. So what I mean by that. Anyway, so don't need to worry about that. So the first thing that one wants to do is remove the genes. So remember, now you have that matrix of 4,000 some genes in your Mycobacterium tuberculosis that I showed you the matrix. The first thing that HR does, or for that matter, any normalization software that you use would do, is remove the genes with zero read counts across all samples. So later on, we are going to count the mean expression of your genes, which you will see later on. What can happen is when you have a mean, you have this term of n, which is the number of total genes. Now, when you include the genes which also have zero read count, so first of all, biologically, it doesn't make sense to include that in the analysis, because the genes which will have zero read counts are not really adding to the analysis in any way, because they are not being expressed in either of the conditions. So you might want to drop those genes. Statistically speaking, these genes which have all the zero read counts will lead to a larger value of n. So uh, that can reduce your mean from the actual mean that you could have gotten with just the genes which would have really been expressed in all these samples. So for that reason, we need to remove these genes with zero read counts, and we'll come to another specific reasons why we did this when I explain to you in the next slides. So for the first step is you remove all the genes with zero read counts before proceeding in the analysis. Second is you choose a reference sample against which all the other samples are normalized. And so let's say I told you that we have 8 samples, right? You have pyruvate, you have glycerol, and in pyruvate and glycerol, you have different pH conditions, the 7 pH 7 and 5.7. Those were the samples in our data set. And for each sample, you have 2 replicates. So you have 8 samples. Now, the idea is to choose a reference sample against which you will be normalizing all the other samples. So this would help us to take care of the bias that might be coming with respect to the differences in the composition of RNA. Now, then the next question is, how does one choose for a reference sample? What could be a bad reference versus a good reference? So imagine there is a sample which has almost all the genes as zero, except 2 or 3 genes which are really being expressed. That would be a bad sample, because um, you want to take the set of genes which would be the most average uh, uh, as compared to all the other samples, and that would then be used to normalize all the reads in all the other samples. So what I mean by that is, let's imagine that you have these 4 samples with 2 conditions each: wild type 1, wild type 2, mutant 1, mutant 2. And let's say these are your read counts that you get, the raw read counts that you get for all these 4 genes. Now, the first thing that Agile would do is it will scale it to the total library size. So 10 plus 20 plus 30 plus 40, the total sequencing depth or the library size for wild type 1 is 100. And then you can similarly count the total reads for wild type 2, mutant 1, mutant 2. Now, these are your total reads. So once you scale this, you will get, this would be the scaled read count for gene A in wild type 1 would be 0.1, 10 divided by 100, and so on and so forth. So we can do this exercise for all our samples. We will scale it by the sequencing depth. Next is that you have to choose the reference sample, remember. So uh, what you would then do is you will calculate the 75th quantile for each sample. So what I mean by 75th quantile is that value. So if you take just wild type 1, the 75th quantile would be that value which contains 3. So in this case, there are only 4 values, right? So the 75% would be 3 out of 4. So you will want to get that value which has 3 or less than 3 values which are smaller than that number. So 0.3, if we take as a 75th quantile, there is only 0.3, 0.2 and 0.1 which are less than 0.3. So this will be your 75th quantile. Similarly, in wild type 2, your 75th quantile would be 0.11. So the only reads that are smaller, the only read counts that are smaller than 0.11 are 0.09 and 0.04. And so you will repeat this exercise for all of your samples. So you calculate the 75th quantile, and then you take the average of these 75th quantile values. So in this case, the 75th quantile average would be 0.216. Now you choose that sample out of your samples, whose 75th quantile is the closest to the average value of 75th quantile. So which is 0.277 in this case. So from now on, mutant 2 would be our reference sample, because what we have found is that this is the most average sample out of our 4 samples. So this is the reference sample, and this is what we are going to now use to one by one normalize it with all the other the samples. Now we have to select genes which would be then used to calculate the scaling factor. Now remember, or you should know, that HR calculates this scaling factor individually for each of these sample pairs. So mutant 2 with wild type 1, mutant 2 with wild type 2, mutant 2 with mutant 1. So it would calculate a scaling factor which would be specific to each of these samples, and does not calculate a common scaling factor. And that's the whole reason why you chose a reference sample which would be the most average of it. So just to segue into the earlier point, just to give that thing a closure, in even quantitative RT-PCR, when you are running, if you want to check the expression of a particular gene through experiment, you also run a control which is usually your housekeeping gene. For example, uh, it could be in eukaryotes, it's usually GAPDH or beta-actin. So the housekeeping gene is something whose expression does not change no matter what the condition is. So because it's an important gene, it needs to be expressed. So the expression of that particular gene does not change. And so that is sort of the idea over here as well. So you are trying to reach to those set of genes whose expression is moderate, or whose expression is not changing across all your conditions. And once you get that sample which is unaffected by the condition no matter what, you can then scale your reads up or down with respect to that set of genes. And we'll come to that, how can that affect. So now you have mutant 2 as the reference sample so far. And now we are going to repeat this exercise of selecting genes to calculate scaling factor for each sample pair respectively. So now this is not a graph, just to be clear. All I'm doing is I'm representing a reference. In this case, our reference is the mutant 2. And let's say we are doing this exercise of selecting genes to calculate the scaling factor for wild type 1. Now, if I tell you there is a gene on this line which is present at this particular point, this means that it is very highly expressed in the reference, but not expressed in the wild type at all. And similarly, if there is a gene which is present in this extreme, it means that it is only expressed in the wild type 1, but not expressed in the reference. So these are your biased genes. One way that we can quantify such genes is by log fold changes. So you can divide. So when I say log fold change, what I mean is, let's say we are talking about, let's say gene A is the one that is biased. It's on the reference side. It's on the wild type 1 side. So what you would do is you will divide 0.1 divided by 0.019 and take a log of it. So that would tell you how much is it overexpressed or underexpressed in either of these conditions. So this is your bias genes that you can take care of, with the log fold change. You can also think of the genes in the other dimension, which is if a particular gene is present over here, this means that it is having a high expression in both the reference and the wild type. So it was being expressed highly in both of these reference and the wild type. On the other extreme, you can have this gene in the lower end, which So this would mean that it has a lower expression in both the reference and the wild type. So what I'm trying to say is that all these 4 genes are on the extremes, and therefore these are definitely affected by the sample that you are dealing with. So these are your so-called bias genes. What HR assumes and wants to get at is that majority of the genes are moderately expressed. So these would be these genes which are not affected by the condition of the sample, and they always have the same expression throughout. Uh, so that's what we want to get at. Now, the way in which you quantify these genes, either highly expressed or lowly expressed in both the samples, is by calculating something like a geometric mean. So if you remember from our mathematics classes, an average arithmetic mean is not robust to outliers. So for example, if I have a sample which has 6 which has 6 readings, let's say the readings are 1, 2, 3, 4, 5, and then suddenly you have 4,000. Now, what arithmetic mean would do, that is sum of all observations upon total number of observations, it would pull your average towards the 4,000 outlier which you had. But geometric mean would be robust to that. What I mean by that is that it would still try to give you a true picture that, you know, my data is actually concentrated in 1, 2, 3, 4, 5, but there is some outlier which might be present. So the geometric mean will give you a more real representation barring the outliers as compared to the arithmetic mean. So if you remember in the first step, when we removed the gene counts with all the 0 reads, one of the major reasons was that the geometric mean can be affected by n. So you would want to reduce your n so that your geometric mean is a truer representation of your data. So that's another reason why we did that. So basically we can calculate this and this using a geometric mean. Agile exactly does not calculate geometric mean, but what we have seen is that when you take, so it calculates something called as a mean of the logs. So it takes the log of this value. So it will take a log of 0.1, then it will take a log of this number 0.019, and then it will take a arithmetic mean. So that would be equivalent to a geometric mean. But the idea was, since usually you are more familiar with the geometric means as compared to the logs, talking in terms of logs, so I gave that example that geometric means are robust to outliers. So that's what it does. So then finally what it does is it will calculate these 2 columns. So remember, we are still talking about this pair of wild type 1 and mutant 2. It would calculate the log fold differences, which remember will take care of these extremes, the horizontal extremes, and it will calculate the mean of the logs, which will take care of the vertical extremes. And after calculating these, it will then sort these. So you sort this column and you sort this column, and you what then you do is you apply the filters of 30% and 5%. So what it will do is, Agile does is, it will sort this column and it will sort this column, and after sorting, whatever top 30% reads you are getting and bottom 30% genes you are getting, the top 30% and bottom 30% genes, it will trim those genes. So it will remove those genes in this, and the cutoff it uses is 30%. For mean of logs, it would remove the top 5% and the bottom 5%, and you will be left with some of these genes. So how they have arrived with this number is, all they say is that empirically they have seen that a 30% cutoff or a 50% cutoff or 5% cutoff works for most of your analysis, but then they have also given an option where you can change these filters. So now remember, all you are doing at this point by trimming these genes is you are getting to this particular set of genes which will not be affected by your samples. That's all you're doing over here. So then after you have those genes, then you will finally calculate the weighted trimmed mean of log to ratios. So the reason why it is called is the TMM normalization, or the trimmed mean of M values. By the way, this is your M. This is your M and this is your A. A is average. So this thing is called as an A, and this is your M. So we will be also doing, when we are doing the agile practical, we will be coming to something called as an MA plot. Then that time we'll discuss this again. So the trimmed mean of M values is basically you are then calculating these log fold differences for each of these genes, and then you are calculating the scaling factor. So now once you have selected these genes, you will then calculate the weighted trimmed mean of log2 ratios. So genes with more reads mapped to them, that is the highly expressed genes, will get more weight. And why is that the reason uh, as compared to the lowly expressed genes? So I'll explain that with this example. Now imagine you have these 4 genes, A, B, and C, D. Now A, B are having a high expression in the wild type as compared to the mutant. When you calculate the log2 ratio, that means 202 divided by 101, and you take a log of that, it will give you 1. Now for gene B, you do the same thing. You have 204 divided by 101, and you take a log 2 of that, so it will give you 1.01. So this means that the log fold difference of gene A in 2 conditions and gene B in 2 conditions is 1 and 1.01. So that means there is approximately twofold change of gene and, A and B. Now, if you don't look at this log2 and if you just look at these C and D genes, these are very low read counts. So you have just 2 in wild type and you have 1 in mutant, and you have 4 in wild type and you have 1 in mutant. You know by your own logic that this is a very low read count, and essentially these are not really being highly or lowly expressed in both of these conditions. But because your reads that are mapping to C and D in both these conditions are very low anyway, and remember that could be because of the RNA composition bias that we had talked about. If you still go ahead and calculate the log2 ratio, because remember at the end you're only dealing with the log2 ratio, you're not really looking at these. What might happen and what it might end up happening is that you are going to see 1 and 2 over here, and you might end up thinking that, oh, gene D is also getting overexpressed as compared to A and B, for example. So therefore you need to give more weight to these genes as compared to the other genes, and by doing so you are then normalizing for this effect, where you can get log2 ratios which could be misleading as compared to the raw read counts which you have got over here. So that's exactly why we have to weigh your genes according to the number of reads that are mapping, and that's exactly what HR also does. And finally, it scales everything around 1. So let's say right now, after doing this weighted average, you had centered the data around the mean of all the scaling factors. So imagine you had calculated the scaling factors for wild type 1, wild type 2, mutant 1, mutant 2, and then you take an average of all the scaling factors. You then shift that scale to 1, so that also helps you further, that we will discuss whenever we reach to that point. So that's the last step it does, and that's how you get a scaling factor. And once you get that scaling factor, you then all you have to do is just whatever read counts you had gotten, you have to just either uh, bump your so-called apparent read count up or low depending on what your sample is. So if let's say there are a more number of highly expressed genes which are being expressed in a particular sample, the scaling factor would be low for that, just to bring that thing down, the effect of highly expressed genes down. And if you have a sample where you have more number of lowly expressed genes, the scaling factor, because you have weighted these trim means, would bump your effective library size to a higher value, just to take care of that RNA composition. So finally, what we have seen is that a raw read count of a gene could be a result of many factors, and you essentially what at least HR does is it normalizes for library size and RNA composition bias, and then finally whatever values you get, those you are getting closer to the abundant values or the real abundance values that you can then use to compare across different genes. So that's all for the theory. I don't think we have time left for. We still have 10 minutes to go. We'll continue with the practical. So we will be starting with the practical and doing this particular step. That's what I had thought of, but I think the theory took time. So we still have 10 minutes for 1 hour to go. So do you guys want to start off that practical or continue it in the next class?
Mohak, I think it's good to stop here, no? And if you
Okay.
do next, because it would that would be otherwise you would be stopping at an odd point.
Yes. Okay.
And I guess there won't be a class this Thursday, right? With the interviews going on.
Yes. Yes.
So next Tuesday.
Yes.
Mohak, could you share these slides or other resources, because I never studied biology in college, and so I'm feeling a little lost. So just a big picture of what all we are trying to do, and maybe just to put it into perspective.
Okay. No problem. But as of now, do you have any specific questions that you would like to ask?
No. So the thing is, if I don't read, I try to understand myself, I won't have specific doubts.
Okay.
So
No
I'll try
problem.
to
But
do that.
okay. Okay.